stuck fermentations is a lack of essential micronutrients. Some of the B complex vitamins are an example, like thiamine, which is vitamin B1, and it's necessary for the metabolism of pyruvic acid, or pyridoxine, which is vitamin B6, is a coenzyme in amino acid metabolism, and niacin, which is part of NAD, which is essential for electron transport and electron transfers. And as I state down at the bottom, these compounds are difficult to measure. There's no rapid test for them, and so they can often be implicated in stuck fermentations. Other things, including stress factors such as temperature and ethanol toxicity, inhibitors, solids, and biological competition, are things that I'll go through in the next slides. The temperature stress, again, is probably the biggest one, and certainly impacts membrane fluidity, and which allows or disallows things to move freely through the membrane. And it also impacts how membrane proteins are inserted and how they function. Temperature stress definitely enhances ethanol toxicity. So you have the two synergistic stresses acting together to really damage yeast. And of course, there's less tolerance if nitrogen is limited, particularly early on in the fermentation, which means that the yeast can't make heat shock proteins. Again, they need the nitrogen to make amino acids, and then they need amino acids to make proteins. And so obviously early nitrogen limitation can really influence that ability to handle shock later on. This fourth bullet point is really important. Temperature is the only factor with an immediate direct mortal effect on the yeast. Temperature stress early in the fermentation can make yeast less tolerant to ethanol stress during late fermentation. So, we often say that an optimal range for a red is like 70 to 85 degrees, and a white is maybe 45 to 60 degrees. And so reds, obviously, we want it to be a little warmer so we can enhance phenolic extraction and so forth. But there can be temperature extremes in the cap that can damage the yeast. And so there are people who think that maybe it's best to run fermentations a little colder to alleviate the likelihood of a problematic fermentation. I should back up. I forgot to mention heat shock proteins. Again, these are just proteins that help stabilize and refold denatured proteins in the yeast cell so that they can adapt to stress tolerance. Some of the yeast temperature requirements, this is an interesting concept. Yeast strains obviously differ in temperature tolerance, and temperature plays an important role during yeast replication. So one thing we always note is that when yeasts are growing at their optimum during, say, log phase, for example, they're at their most sensitive when they're undergoing that rapid cell division. And of course, there's an issue with early nitrogen stress, as I mentioned previously. Early nitrogen stress means the cells are unable to make sufficient amounts of heat shock proteins that would protect the cell. The more yeasts are stressed early in fermentation, the lower the peak temperature they can tolerate. So again, that's just an adaptation aspect of it. A little bit about ethanol toxicity. Ethanol toxicity is something that typically impacts membrane composition. So changes in composition can affect function, as we know. So we're talking about transporters and things like that can be damaged, and therefore the cell can't bring in hexoses as well. But yeast respond to increased ethanol concentrations by producing their version of cholesterol, which I've mentioned previously, ergosterol or ergosterol, depending on who you talk to. But ergosterol is probably the right way to say it. Anyway, but many times the yeast will vary their membrane proton ATPases, for example, or decrease the numbers of them, because when the membrane is in such disarray, the cell becomes more leaky to protons, and they really don't want to just vomit protons out into the world, because the protons are essential for transport. They're essential for making ATP and so forth. And so the cell is just willy-nilly leaking protons. It can be very detrimental to the cell. Of course, ethanol has a direct effect on the phospholipid bilayer, disrupting integral membrane protein function. And so the goal is to decrease interdigitation and enhance membrane fluidity. And so ethanol stress can increase this concept called interdigitation. So you see here's a normal membrane, but then high ethanol results in this collapsing. You see this here of the membrane into what's called an interdigitized membrane, and so that membrane becomes essentially functionless, and the cell is now under stress, unable to transport. The membrane is not fluid when it's like that. Increases in the passive proton flux into the cell, causing stress to maintain cellular pH, and that's a big issue. So that's why I said here, the cells, they don't want to pump out protons because obviously they need them to make energy and do transport, but they also don't want a ton of protons coming back into the cell, which would, in a sense, change the pH, essentially lower it, creating a more acidic environment that the yeast then has to balance out. The presence of inhibitory compounds is also a huge issue in problematic fermentations, and so usually it's inhibitors produced by other microbes. So if you get a bloom of acetic acid bacteria or some lactic acid bacteria, those two acids can certainly inhibit yeasts. But other interesting ones, like inhibitors produced by the plants that came in with the grapes, like phytoalexins or PR proteins, both of those are pathogenesis-related proteins that the vines will use to prevent fungal infections and infections by other parasites. And certainly pesticides, if they get into if they come in with the grapes as well, those chemicals can often be inhibitory to your Enococcus and malolactic or even your primary yeast. Certainly fungicides, that's not a good one. So if you have fungicides that are coming in from the vines as well and on the grapes that are not sufficiently washed off, fungicides that are trying to minimize growth of yeasts and molds and things out in the field that come into the fermentation will be a huge problem. One of the other interesting ones that's picking up steam in terms of research is quorum sensing compounds. We know that, for example, Candida species will produce a compound called farnesol, which can actually induce apoptosis in Saccharomyces, which apoptosis is just a fancy word for programmed cell death. And so again, you've got these microbes producing higher alcohols like farnesol and so forth that can actually kill your desired yeast. Physical stress is an interesting one as well, and it basically just refers to insufficient solids. Usually this occurs in over-clarified juices, and they might initiate more slowly. And usually some people want to do this to reduce the solids, to reduce the need for fining or filtration on the back end. And so it's not an uncommon practice, but it can lead to problematic fermentation. And one of the reasons is, when you're clarifying, soluble pectins that would keep the solid suspended are eliminated. And so again, these soluble pectins keeping solids suspended provides yeast binding sites so that they can sort of collect on these surfaces, and that almost forms a tabletop on which they can ferment and communicate and actively metabolize. Another big one is obviously biological stress. And so, again, we're talking about competition for nutrients with wild yeasts and bacteria. A lot of times the wild yeasts can produce killer factors that can inhibit or kill your desired yeast. And again, I'll talk about those at another time. And certainly bacteria that can gobble up sugars and belch out acids and alcohols and so forth can also contribute to the stress on the yeasts. And one of the other cool things that bacteria produce are small antimicrobial peptides that they use to sort of antagonize each other. And I think, Enococcus strains that are used for malolactic are often selected for that have these small antimicrobial peptides, because that gives you an extra boost. You've got the malolactic, but you've also got a malolactic that can produce these antimicrobial peptides to sort of knock down competition. But sometimes the wild bacteria produce these things, and these antimicrobial peptides. They're called bacteriocins, and these bacteriocins can again affect the desired bacteria in your fermentation, particularly the malolactics. But like I said, preferably we want the malolactics to be producing the bacteriocins and killing other guys. Another thing that yeasts have, which is really weird, is a prion type phenomenon. And we know that prions are, they can cause mad cow disease in cows, but we could also get mad yeast. They obviously don't behave the same way. But anyway, what a prion is basically a protein that exists in a certain form that can catalyze the alteration to a different form of other proteins around it. And so, prions form when a protein changes into a stable alternate conformation. So again, a prion protein can catalyze the conversion of other proteins around it to a different form. And so spontaneous prion formation can occur in response to cellular stresses like heat. So you can imagine if this is an essential conformation for a certain phenotype, and if this thing then spontaneously triggers other proteins around it to change into this new conformation, then these proteins collectively would have a different function. And so one example is this GAR plus or GAR plus phenomenon. This is a prion that is resistant to glucose-associated repression. So what that means is that it acts to de-repress non-preferred sugar transport. So to back up a little bit, the yeasts will preferentially take in glucose, or that particular hexose, and once they've consumed that particular sugar, then they switch to using non-preferred sugar sources. And so that's an adaptation. Obviously you want to eat the best things first and quickest, so that you can then move on, and then you can move on to eating the less desirable things. And so what GAR plus prion does is it essentially de-represses that. It means that the yeasts are no longer limited in what sugars they can eat in the presence of glucose. And so we think that that's a bet hedging sort of evolutionary steps that allows the yeast to be rapidly changing in a fluctuating environment, meaning changing in its diet preferences in a rapidly fluctuating environment. Evidence indicates it can be induced by stress early in the fermentation, and certain bacteria might provoke prions to start converting other proteins to different conformations, such as Pediococcus and Acetobacter.